Hi, I'm Tanja. The research discussed in this video are our studies of the host immune response and its reaction to the presence of the Borrelia burgdorferi bacterium. One of the things we have focused on are how different hosts react to the pathogen and why some get inflammation while others don't. Studying these mechanisms will help us understand the pathology of Lyme disease. One observation is that wild mice, which are carriers of the organisms or reservoirs, do not exhibit any symptoms despite having many bacteria in their tissues. This makes sense, as a mouse with Lyme symptoms would not be able to survive in the wild. So how is their immune response different from that of other mice or humans?
Humans develop symptoms with infection to B. burgdorferi. The symptoms they develop are the result of the immune system recognizing the bacteria and trying to kill them by activating inflammatory responses. Killing the bacteria with antibiotics gets rid of symptoms in most patients, but even without antibiotics, all the symptoms of Lyme disease will resolve over time. Part of this is because the immune system does control the infection to a degree, but it is thought that there is still bacteria present in the tissues. This means that over time, similar to wild mice, the human immune system stops responding to the presence of the organism.
So let's start with the mouse reservoir host.
In a healthy, uninfected mouse, the immune system is just sitting around in an inactive state, waiting to recognize an invader and begin a response. All the blue cells are in a resting state. If the mouse gets infected with a pathogen that causes disease, the immune cells come in contact with the bacteria and become activated, releasing signals to call for help. With the Lyme bacterium, the organisms get into the tissue, but no distress signals are sent. The cells and tissue behave just like the uninfected mice.
So why isn't he sick?
We think the answer is tolerance. The easiest way to think about this concept is like traffic lights. Immune cells, like macrophage, neutrophils, and dendritic cells, act as the traffic light, assessing danger and signaling stop or go to invading bacteria. They are the sentinels, sampling the environment and deciding whether to sound an alarm or not. If the bacteria prove to be dangerous, a real battle between the immune cells and the bacteria begins. The immune cells surround the pathogen to kill it and release inflammatory mediators, recruiting other immune cells and causing inflammation. If after an initial sampling during a slow yellow sign, the bacteria prove to be harmless, the immune cells retreat and the bacteria get the green light to proceed. This happens anytime these same bacteria are seen again, a mechanism we refer to as tolerance. This type of effect is applied to good bacteria in our intestines, the commensal microbiota. We think that Borrelia burgdorferi has taken advantage of mechanisms used for preventing responses to good bacteria for developing tolerance. This allows it to establish a long-lasting infection. We think that tolerance is eventually what causes the symptoms to go away in most people, even if you don't treat the infection. But what if the mechanisms by which we generate tolerance go wrong? It is possible then that those patients continue to treat the bacteria as a dangerous invader, leading to inflammation and prolonged symptoms.
So what are we doing in the lab?
Our studies focus on how the immune system signals and controls this tolerance to the pathogen. Our long-term goal is to exploit the immune system to alleviate symptoms in patients who don't respond well to treatment. We are looking at a variety of contributing factors such as immune signaling, metabolism, cell-to-cell interactions, and regulation of gene expression.